Okay, this is getting into the microbiology of milk, and we can talk more about this during cheese making, but we already covered this principle. Where is milk microflora coming from? It's not coming from the inside of the cow, it's kind of coming from the outside of the cow, so the feed, the air, the manure, it all, that's where it happens. And then, you know, that you'll never see a cow's udder looking that clean. This is like a photoshopped, beautiful, just scrubbed 4-H cow in the picture. Usually there's some amount of debris going on the outside of a cow, especially when they're on pasture. But that is what's going to influence milk microbiology. And then water that is used to clean the equipment and the cows in the barn can also bring microbes with it that don't tend to be, they're either sort of neutral, but there's one called Pseudomonas, which isn't pathogenic. It doesn't hurt anybody, but it causes off flavors in cheese and kind of like a fluorescent smear where it should be a nicer color. You'll see kind of, it looks like a highlighter on the cheese. And so you have to have equipment that's really well cleanable and with really hot, you know, sanitation cycles, because if milk is hanging out in lines, microbiology, you know, microflora tends to multiply, and not in a controlled way that tends to benefit cheese making. So that's just the point of this slide. Okay, milk quality, this is how farmers get paid. So they get paid based on the amount of microbes in their milk, and that's what the top is about. We talked about components along the butter, along the bottom, the butter. So how much butter, protein, lactose is in the milk are all ways they can get paid. Usually, these are some standards of the milk truck. Somatic cell counts are white blood cells, and that indicates if you have cows with udder infections or sick cows in your herd, or goats or sheep. So you kind of track your baseline somatic cell count, and if you see spikes, you know, you might have some cows that are developing udder infections, so that's important to keep an eye on. Minerals we talked about is something that you can measure. And then the milk urea nitrogen, if the cows aren't getting enough starch or energy in their diet, that number is going to go up. They're going to have partial proteins and basically ammonia in the milk, okay? But the bacterial count is interesting. So basically, this is just a count of how many colony-forming units there are. From the standard plate count is just from the fresh milk. The PI count is after refrigeration. The pasteurized count is after heating the milk. You're just plating up some milk and seeing how many colonies form. And they could be good colonies of lactic acid bacteria that are going to help your milk coagulate and then make good flavors later, or they could be bad guys, or they could be neutral. It's non-discriminatory. It's just how much bacteria is in the milk, which is, to a milk truck, a measure of how much kind of contamination there was at milking. Then they look for specific pathogens like E. coli and Staph aureus, and they look for the category of where E. coli lives, which is in coliforms, which tends to be an indicator of how much manure got into the milk, essentially. So they look at these things as just like hygiene. So there's hygiene and then there's components. And for a cheesemaker, you want a raw milk that's very rich in specific kinds of microbes. So it's important here just to know, you know, the goals that dairy farmers are aiming for, but also a cheesemaking farmer has an extra layer of complexity in that they want a rich microflora in their milk. They want a lot of diversity, but they want, you know, certain kinds, you know, positive bacteria. So, you know, 99% of all, 99.9% of bacteria out there that could be in milk are either neutral or positive, and there's just a few pathogens of concern that are sort of tracked. So it's a kind of a different way of looking at milk quality, just in terms of how many colony-forming units there are. You could have milk chock full of lactic acid bacteria. That's a positive thing. All right, so we can kind of wrap it up here. We'll talk about the types. This is a classification for cultures, which I think we can talk about during the cheese-making portion, and as well as the role of lactic acid bacteria. These are all playing right into the cheese-making portion of the conversation. So I'm going to pause it here to talk about at the end of the milk. Pasteurization is the last thing we'll talk about, which is, you know, you've created this milk with your whatever balance of microbes, and if you're pasteurizing that milk and selling it to the, you know, bottled milk factory, or if it's going into pasteurized cheese, then you're probably slightly less concerned with the exact balance of microbes that you've got going on. So there's different types of pasteurization. You can have a gentle vat pasteurization, so bringing milk just up above the danger zone, right? If you've taken food safety classes, 40 to 140 is where microbes are the happiest, and so when you pasteurize, you bring milk up above 140 to 145. And this is the lowest temperature that you can pasteurize to, vat pasteurization, and it takes a longer time. So if you're a larger facility and you have more sophisticated equipment and you're trying to make multiple batches of cheese a day, you're probably going to be doing HTST, which is a higher temperature. You're going up to 160, but only for 15 seconds. And so that's usually more expensive equipment, but it's a lot faster, not only to bring the milk up to temp, but then to the holding temp time. So artisan cheesemakers tend to vat pasteurize, and then in larger facilities you're going to be doing HTST. Then you can do even more sophisticated equipment does higher heat, shorter time, so really warm, really hot temperatures, 191 for just a second. And then you have ultra pasteurization, which is not suitable for cheese making. So you're basically bringing milk up above boiling, but under pressure, so it never boils for 2 seconds. And that essentially sterilizes milk, and then you have shelf-stable little creamers and things like that. But that messes with the proteins to a place where they won't coagulate normally, and you can't use them for cheese. In Europe, they have kind of an in-between to a raw milk and a pasteurized milk called thermized or heat-treated. In this country, if you heat-treat your milk, you just call it unpasteurized instead of raw. And because you're not technically, it's not raw milk because you have heat-treated it. The Raw Milk Cheesemakers Association requests that you don't call it raw milk. So on our labels where the cheese is heat-treated before cheese making, but it's not legally pasteurized because it doesn't get up to 145 for 30 minutes. It might only get up there for a few seconds. We call those unpasteurized. So we have raw milk cheeses, pasteurized cheeses, and unpasteurized cheeses or heat-treated cheeses in our collection. So the U.S. government doesn't recognize thermized, but there are standards of what thermized means in Europe, for instance. And then, of course, raw milk, the standard is you're not supposed to bring milk above the body temperature of an animal, so you're not supposed to adjust the temperature higher than, if you refrigerated your milk, you can bring the milk temperature back up to, the temperature it would have been when you milked the animal, which is usually kind of the start temperature for cheese making. But the point is you're not supposed to treat the milk with heat before you start to make the recipe. So after you add your cultures and run it, if your recipe calls for heating the curd or cooking the curd to a hot temperature, that's okay because that's part of the cheese making process, and you've already involved microbes that were present before that step. So that's the nitpicky raw milk rule there. All right, so that about does it for part one. Thanks for tuning in, guys.